Hello students, welcome to EPG Pathshala. I am Dr. Manoj Kumar from Department of Biophysics, All India Institute of Medical Sciences. Today we are going to talk about Module Molecular Dynamics Simulation Part 2 under the paper Bioinformatics. In this module we will discuss about the analysis of molecular dynamics trajectory produced during MD simulation and applications of molecular dynamic simulations, followed by the special types of molecular dynamic simulations. Students, this module is continuation of previous module, Molecular Dynamic Simulation Part 1. In previous module, I have discussed about the basic features of MD simulation, requirements for MD simulations and setting up MD simulation run. I would like to refresh you briefly about the salient features and points discussed in the previous module. Molecular dynamics is a computer simulation method. It is used to study the dynamic behavior of molecules like protein, nucleic acid or small molecules. MD simulation or molecular dynamic simulation is a computer simulation technique which is used to simulate the motion of particles. It is based on Newton's second law of motion, which states that force is rate of change of momentum. This molecular dynamics simulations technique is used to study the dynamic behavior of biomolecules like proteins. Molecular dynamics simulation is based on Newton's second law of motion, which states that rate of change of momentum determines force. That means force is equal to mass into acceleration. So if we know the force is acting on a molecule of known mass m, then we calculate the acceleration. From acceleration we can derive velocity, and after that we can calculate the position of the molecules with respect to time. Let's apply this principle for simulation of protein. Since we know the mass of each atom in the protein and also charges on them, so we can calculate the forces and acceleration, velocity and position of atom with respect to time. Foundation of molecular dynamics simulation comes from work of Maxwell, Boltzmann and Gibbs in the area of statistical mechanics. Concept of ergodic hypothesis from statistical mechanics states that the time average of trajectory is equivalent to ensemble average. That means macroscopic properties like temperature, pressure, heat capacity can be derived from microscopic data like time average of trajectory of a single molecule or a very limited number of molecules. This principle helps to derive important thermodynamics information from trajectory. We have learned in the previous module that force fields are soul of any computer simulation program. Force fields are generally interaction potential which has been derived from set of parameters like atomic radii, charges, bonding, hybridization states, etc. Interaction potential can be bonded like bond stretching, bond angle bending and torsional potential, or non-bonded like electrostatics or van der Waals potentials. Molecular mechanics force fields are empirical in nature and they have been derived from parameterization or fitting of experimentally observed or through precise quantum calculation potentials to match the actual potential energy or potential energy surface of the molecule. That's why we say it as an empirical force field. For a simulation to run, force fields are assigned to each atom of the molecule. The most commonly used force fields of protein are AMBER, CHARM-M and GROMACS. Setting up MD simulations. These are the steps which has to be followed for simulations of a molecule. Preparation of molecule and then setting up the MD run parameters, followed by simulations in 3 stages, and finally the analysis of the result trajectory. As we have seen in the previous module how to set up MD simulation run. First we need to prepare the molecule which has to be simulated. That means the MD simulations of a protein has to be performed before the start of MD simulations. Like the initial conformation of molecules which has to be simulated must be in lower energy conformation, and also they must have the correct stereochemical parameters and electrostatic parameters as well, like protonation states and partial charges, because we know that all the calculations are performed based on the partial charges. proteins should be also in the appropriate solvation environment. That means we need to solvate the protein in their native environment so that we can study the dynamic behavior as close to reality. Then we need to set up the appropriate run parameters of MD, like what type of ensembles we need, where NBT or NPT, that means canonical or microcanonical, isothermal, isobaric. Also we need to set up the thermostat and barostat, which is appropriate for the objective of MD simulations, like whether we need to put Benson temperature bath or Newhover method. In case of barostats also, we need to set those parameters according to our objective. We also need to perform the MD simulations for a time period or time scale which is required to cover the property of that molecule. That means whether we have to run the MD simulations for nanoseconds, microseconds or milliseconds. If we want to perform the MD simulations for the refinement purpose of a protein structures, then we run it for hardly 10 nanoseconds. But if we need to study the side chain movement, loop movement, then we need to run it for the longer duration like microsecond. Similarly, if we need to study the protein folding, then we need to run it for millisecond to many seconds. We also need to fix the time step, the step size after which the calculations is performed in MD simulations. Similarly, we also need to put the right integrator to derive the motion of the particles. MD run can be categorized mainly in 3 stages like initialization, equilibration and production. Initialization means the random velocity is assigned to each atom of the molecule at the start of simulations to start the movement of the molecule. Equilibration stage is followed by equilibration process. So equilibration stage corresponds to duration of MD run when the simulation system equilibrates its properties like normal distribution of velocity of the system. That means the system achieves the equilibrium state and negates the external force that we have assigned earlier to move the molecule. Pressure, temperature and energy parameters during MD simulation is also used as a measure to assess the equilibration process during simulation. Third and final step, which is useful for the analysis of the further process of simulation, is the production stage. Only trajectory corresponding to this production phase should be considered for further analysis and used to calculate the microscopic properties like free energy of binding or the changes in structures of the protein. So we must keep this point in mind during analysis of MD trajectory. MD trajectory is the main output file in a MD run. It contains the informations about all the conformation of the molecules which has been generated and saved, like coordinates, velocity and energy parameters of all the conformations. From trajectory, we can visualize the movement of molecules at atomic resolution and femtosecond time scale. Output file of any MD run includes trajectory file, energy file, coordinate file, topology file and log file. But it is the MD file which is more important. MD trajectory file is saved in various formats like .xtc, .tpr in case of GROMACS simulations. However, it can be written in different extension in different simulation program. But everywhere, trajectory file contains about informations of coordinates, velocity, energy, temperature, pressure of all the conformations saved during MD simulation. We analyze this file to derive required information resulting from simulations process. We can visualize the motion of each atoms of molecules at femtosecond to picosecond scale using appropriate program. Each simulation engine has its own trajectory and arranger visualization program. We can run the trajectory from GROMACS in VMD tool. We analyze the following parameters or property during MD run from the MD trajectory file, like temperature, pressure, volume, energy. Besides these parameters, we also analyze the root mean square deviation, fluctuation, secondary structure analysis for protein, radius of gyration, hydrogen bond analysis, covariance, protein-ligand interactions. We analyze the different parameters from trajectory file. like temperature, pressure, volume, density, potential energy, kinetic energy, and total energy. These parameters are generally used to locate the equilibration point of MD simulation. It also indicates the stability or disturbances during MD run. Other important parameters are analyzed using coordinates of molecule with respect to time, that is, the different conformations of the molecule saved during MD simulation. Root mean square deviation and root mean square fluctuation, radius of gyration, secondary structure pattern, hydrogen bond pattern can be analyzed in case of protein, and also protein-ligand interactions can be analyzed in case of protein-ligand complexes. The root mean square deviation of protein can also be used to identify the equilibration phase. However, it is not the right parameter to indicate equilibration state in case of highly mobile domain containing proteins like kinases. This is a typical parameter plot of MD simulation run. It has to be analyzed for equilibration pattern and stability of MD run. This is a 10.2 nanosecond MD run graph of DNA guided ciprofloxacin complex run on Desmond simulation program. Properties under observation in this case includes volume, temperature, pressure, potential energy, kinetic energy, and total energy of the simulation system. This graph clearly indicates that the guided ciprofloxacin complex has achieved the equilibration state very quickly at about 0.2 nanosecond. And this may be due to reasonable low energy conformation of the complex at the start of simulation. There is also not much variation in any of the following parameters during simulation. Minor variations seen in the plot are very small and have a repeating pattern indicating a stable trajectory. Analysis of MD trajectory in terms of root mean square deviation. Root mean square deviation or RMS deviation is the measure of difference between two different conformations of the molecule or two different structures of comparable size and fold. It is done by superimposing the conformations and then calculating the square of the difference between each atom pair, and finally taking their square root to arrive at root mean square deviation. In case of MD simulation, RMS deviation indicates the difference between two conformations of the same molecule with respect to time. It gives a measure of stability of a structure during MD simulation. That means lesser the variation, more stable is the structure, and vice versa. We can calculate the RMS deviation for all atoms or only a selected group of atoms in a molecule which has to be simulated. Like in case of protein, we can calculate the RMS deviation for only backbone of the protein or side chain of the proteins or total atoms of the proteins. This diagram represents the root mean square deviation of different conformations of protein during simulation run of 10 nanosecond. 3 line indicates 3 different group of atoms of the same molecule. Like red color line in the middle is showing the deviation between all the atoms of the protein including backbone and side chain atoms with corresponding conformations with respect to time, while the lower line in blue indicates deviation for only backbone atoms and upper line in green denotes deviation only for side chain atoms of the protein. This also reveals that backbone of the protein is more rigid than side chain conformations of the protein. This happens because of stable configuration of protein backbone due to partial double bond character of peptide bond and planarity of the peptide bond between CO and NH group, while side chain torsions, chi torsions, are more flexible compared to backbone protein torsion phi and psi. This is another example of RMS deviation calculations used for other purpose. This graph indicates the variation in protein structure during simulation at different temperatures. This reflects that variations are higher at higher temperature than lower temperature. This happens due to higher kinetic energy of molecules at higher temperature. This principle can be used to study the temperature-based stability of protein. Theoretically, we can find the optimum temperature range of any proteins through temperature-based simulations. Similarly, RMS deviation plot of wild-type and mutant protein during MD simulations can indicate the stabilizing or destabilizing effect of mutation on protein structure. In this case, we can see that RMS deviation in mutant protein is higher than wild-type protein. This indicates the destabilizing effect of mutation on protein structure. If we have more than one mutant form of protein, we can predict the degree of destabilizing and stabilizing effect of each mutation. That means we can predict which mutation could be more deleterious among them. In this way, we can see that mutant 2 is more damaging than mutant 1 and 3. This can be interpolated with the function of protein also. This is very commonly used and analyzed properties from MD trajectory. Similarly, we can plot the root mean square deviation of protein and ligands for molecular dynamics trajectory of protein-ligand complex. This will indicate the stability of binding of ligands. This is also indicating the induced fit mechanism during protein-ligand binding, as we can see the change in RMS deviation in case of protein as well as ligand. Ligand is found to be more mobile than protein, as here it is indicated in the graph, where ligand has higher RMS deviation as compared to protein. Root mean square fluctuations. It is another analyzing parameters used to analyze the MD trajectory in addition to root mean square fluctuation, especially in case of protein simulation. It is measure of average movement of position of atoms with respect to time. This is calculated by computing the positional displacement of atom between different conformations at different times and then averaged by number of conformations. It is averaged over time for individual atoms or residues in contrast to RMS deviation, which is averaged over all the atoms and gives time specific values. It gives the idea of movement of a specific region of protein with respect to overall structure. This is a typical RMS graph. This graph plots the root mean square fluctuations calculated from MD run of 10 nanosecond for a protein of 200 residues. RMS fluctuation has been calculated for individual amino acid residues over all the saved conformations for 10 nanosecond MD run. This plot indicates that two regions of protein, one near residue 50 and other near residue 150, has higher mobility than other regions of the protein. This might be due to more flexible loop in that region or a more mobile motif or domain. Like in case of kinases, this may indicate towards the lead motif and P loop. This is an example of MD simulation of a kinase protein, which has two very mobile domain, lead and NMP, while core domain is more stable. From A and B plot, which shows the root mean square deviation of different domains. From this domain, we can see that the lead and NMP domain are more mobile, while core domain is less mobile or more rigid. That is indicated by their respective root mean square deviation. In D plot, we can see the root mean square fluctuations of the same MD run. And we can see here that the residues between 40 to 70 and residues between 120 to 160 are showing more fluctuations. And those corresponds to nucleoside binding domain and lead domain, which are more flexible or movable domain in kinases. We can also analyze the secondary structure conformation of proteins during different time scale of MD simulations. This is the figure which indicates the MD simulations of DNA gyrase, which is composed of two different monomers and consisting of 1200 residues. This graph plot of secondary structure pattern of protein depicts the secondary structure conformation of residues during different time point over 10 nanosecond simulation. Red color region indicates the helical conformations, while cyan color indicates the beta sheet conformation, while the other regions indicates the irregular loop conformation. Hence, this plot can give the idea of secondary structure composition and also the secondary structure conformational stability of each region during MD simulation. This plot can also give the idea of percentage composition of secondary structure of protein, which may vary during course of simulation. Hence, this can be used to study the change in secondary structure composition by simulation at different temperatures for any set of proteins. Radius of gyration is an important parameter which is analyzed for MD trajectory. It gives the idea of distribution of atoms in terms of mass in protein under simulation. It is measure of compactness of protein during simulation process. It can be calculated for different mutant form of proteins to study the effect of mutation. The smaller the radius of gyration, more compact is the structure and vice versa. So if we have radius of gyration for wild type and mutant form of protein, then we can see if radius of gyration is increasing, that indicates the destabilizing effect on protein. Principal component analysis of MD trajectory is also very often performed. Principal component analysis is a multivariate statistical method which transforms the series of coordinated observation into a set of orthogonal vectors called principal components. This is required to remove the random thermal motion, which is translational or rotational motion, so that we can capture the actual motion of the protein, which are more significant. It helps to determine the most significant motion in dynamics trajectory. It also helps to determine the domain motion in a protein with respect to the other domain. Molecular dynamics simulations has many important applications in structural biology. It is an important computational tool for study of biomolecules, especially proteins and their complexes with ligands. It is used to study both the kinetic properties as well as thermodynamic properties of the molecules. Using kinetic properties, we can study the dynamic behavior of proteins, like protein folding mechanism, ion channels and transporters opening and closing, refinement of protein structures or their homology models. While using thermodynamic properties, we can calculate the free energy of binding between protein structure and ligands. By simulating the small molecules or liquids, we can calculate their boiling point, pressure, etc. As we have discussed in case of root mean square deviation and root mean square fluctuation and radius of gyration, that using those analysis we can study the flexibility and stability of proteins. This diagram represents refinement of protein structure of molecular dynamics simulation trajectory. The refinement of MD simulation removes the strains in structure in contrast to energy minimization program, which can only remove steric classes. Also, MD simulations achieves the closer to global energy minima state of the protein, while energy minimization program can only achieve local minima. Here we can see the movement of atoms leading to better stereochemical and energy state of the molecule. MD refinement also validates the local regions of stability, like stability and conformation of secondary structures in a protein. Hence, it is very often used to refine the homology model of protein structures. Similarly, we can also perform the conformational analysis of peptides or small molecules where experimentally determined structures are not available. We can identify the most common conformation of small peptides, which can be further used for binding or docking study. Here we can see the different conformations of a small peptide generated during MD simulation. We need to energy minimize each conformations and cluster them using superimposition matrix to find the major cluster, which indicates the most probable mode of conformations. And we can select the lowest energy conformation among the largest cluster as the most feasible structures. Another major application of MD simulation in life sciences is to study the folding of protein. Protein folding is a major area of research in protein science. Molecular dynamics simulation helps to understand the mechanism of protein folding based on thermodynamics properties. However, it should be kept in mind that only simple protein not requiring assisting proteins like proteins involved in protein folding chaperones can be studied for their folding pattern. In this example, we can see the complete pathway of protein folding of a small protein which has been performed by Vincent group. Here we can see how from a linear peptides the secondary structures are formed, beta sheets, alpha helix, and finally how does it fold and stabilizes the protein structure. Similarly, we can also study the opening and closing of ion channel proteins or transporter proteins like glucose transporter. This also follows the same principle of dynamic behavior of protein based on its intrinsic properties like distribution of atom types in terms of charges and masses. This adapted simulation shows the mechanism of opening and closing of potassium ion channel through formation and breakage of different interactions during course of MD simulation evolution. We can also analyze the protein-ligand interactions using MD simulation. We can identify the interactions retained or lost during course of simulation. We can also study the effect of water solvents on the binding of ligands into the protein, including the water-mediated interactions. That is not possible in the absence of water molecules. Hence, we can identify the stable and important interactions between protein and ligands. This will help us to modify the compounds to improve their binding. One important application of MD simulation is estimation of free energy of binding of ligands. This is done by many methods like free energy perturbation, thermodynamic integration, linear interaction energy, and molecular mechanics based Poisson-Boltzmann surface area methods and molecular mechanics based GBSA method. Free energy perturbation and thermodynamic integration methods are time intensive, but they are more accurate, while molecular mechanics based PBSA and GBSA methods are faster but less accurate because PBSA and GBSA methods are using implicit solvent models. So the solvent effect, solvation-dissolution energy cannot be calculated accurately, while free energy perturbation and thermodynamic integration is calculated by performing the MD simulation in the presence of solvent models. That's why they are more accurate to calculate the effect of solvation-dissolution on binding. There are few specialized or modified form of molecular dynamic simulations. They have been developed for special purposes. This includes the simulated annealing molecular dynamics, targeted molecular dynamics, steered molecular dynamics, replica exchange, Langevin dynamics, and Brownian dynamics. Simulated annealing molecular dynamics. It is a MD simulation where molecular dynamics is performed at different temperature for a fixed duration in a single run. Since we are using the different temperatures, so we need thermostat to regulate the temperature. The idea here is to raise the temperature to very high degree like 400 to 1000 Kelvin so that the molecule can cross the conformational potential energy barrier and it followed by the gradual cooling to normal temperature. This approach avoids the chances of molecules stuck in local energy minima. This is more effective conformational sampling technique used to explore the conformational space of the molecule as compared to conventional molecular dynamics at room temperature. Targeted molecular dynamics is a special molecular dynamics tool. This is used to explore the conformational path between initial and final conformation of the molecule. In this case, a biased force or potential is applied, which propels the molecule towards final conformations and negates the effect of random thermal motion. Hence, it helps to speed up the conformational changes to achieve its final state at faster rate than conventional molecular dynamics approach. It is useful for simulation of molecular processes which require larger sampling time, like opening and closing of ion channels or transporters or large movement of domains in kinases. It is a type of steered dynamics and hence a non-equilibrium dynamics and cannot be used to calculate the equilibrium properties like free energy of binding. Steered molecular dynamics. This is similar to targeted molecular dynamics, which is used to accelerate the sampling process with the help of a pulling force. It is used to study the mechanism of association-dissociation of ligands with receptor. Here we can see the process of binding of ligands to the receptor. It can also give the measure of relative binding strength of ligands by calculating the rupture force, which is applied restraint force to steer the process. Larger the rupture force, higher is the binding affinity and vice versa. Hence, it has gained renewed interest in the area of drug design. It can also be used to study the folding-unfolding process of protein and transport of small molecules, similar to targeted molecular dynamics. It is an in silico alternative to atomic force microscopy. So students, let's now summarize what we have learned in this module. The MD trajectory mainly contains coordinates, velocity and energy of each conformation which has been saved during the MD simulation process. Analysis of MD trajectory is crucial for deriving the kinetic and thermodynamic behavior of the molecules which has to be simulated. Similarly, stability and equilibration of MD run is verified by temperature, pressure and energy plot from the MD run trajectory. The MD trajectory provides informations besides temperature, pressure and volume. We also find root mean square deviation, root mean square fluctuation, radius of gyration and other thermodynamic properties like free energy of binding and heat of vaporization of the system under simulations. There are also some specialized or modified form of MD simulation like targeted molecular dynamics, STR molecular dynamics, Langevin dynamics. They have been developed for a special purpose